Lung cancer is a massive problem around the world. It's the most common cancer because it affects men and women, up there with breast and prostate cancers. And unfortunately, it happens in patients who are older, often with many comorbidities. And these factors, combined with the fact that patients often present with quite advanced disease, means that the longer-term outcomes are very poor, with a single-figure survival beyond 5 years, really, across most patients. About 20 years ago now, it was established using combination chemotherapy was better than just best supportive care for patients with advanced non-small cell lung cancer. And for the next 10 years or so, a series of studies, including thousands of patients, compared different platinum combinations and were able to demonstrate very little clinically meaningful difference between them. During that time, around about sort of 10 years ago, it was recognised that there were different populations within that larger non-small cell lung cancer patient population, which had different molecular characteristics. And the first one recognised was EGFR. And a proportion of patients, varying actually around the world, but within Western populations around about 10 to 15%, have activating mutations of EGFR. And it was recognised clinically that the patients who are most likely to get benefit from EGFR targeting drugs had a certain characteristics, and they were more likely to be of Asian descent, to be women, to be never or light smokers, to be adenocarcinomas. And so a very important pivotal study was the IPAS study. This study was performed only in patients with those clinical characteristics and randomized them between carboplatin and paclitaxel, one of the platinum doublet combinations, with gefitinib, one of the first generation of EGFR inhibitors, with a primary endpoint looking at, you can see here, non-inferiority per progression-free survival, but also with a number of different subpopulations. And although the data overall did show non-inferiority, the really interesting information was in the subpopulation looking at EGFR mutation. And here you can see on the graph on the right that those patients who had an EGFR mutation had a significant improvement in their progression-free survival with a hazard ratio of under 0.5. Also quite important to note is the fact that even though these patients were selected to have all the clinical characteristics that made us think they would respond well to these drugs, actually if they didn't have the underlying biological driver, the EGFR mutation, they did dramatically worse with a targeted drug with a hazard ratio of nearly 3. So this is a really important message that actually our clinical thoughts about which drugs might be useful aren't really good enough in this setting where we really need to understand the molecular biology in order to select the right treatment for our patients. So as a translational oncologist, it's particularly interesting to me that sadly, although these drugs were very effective, the patients all went on to develop recurrent disease and die of their disease. But what happened was the patient's tumours were sampled, and in order to understand the mechanisms of resistance to the drugs, the tumours were characterised again, and it was found that in more than half of the patients, tumours harboured a secondary mutation called the T790M mutation. And this biological understanding then went to the chemists, and the chemists developed new drugs which were developed specifically for mutated EGFR and with activity with this T790M secondary mutation. The drugs then went into the clinic, and osimertinib was licensed recently in this second-line setting, basically on 2 single-arm studies, so a phase 1 study and a phase 2 study, small numbers of patients. And you can see here in the waterfall plots, almost all patients who harboured the T790M mutation had a response to treatment, and this treatment response was durable, leading to the license of the drug. This license happened within less than 5 years of the first-in-human studies, which really contrasts with the more than 10 years that it took for the development of the first generation of EGFR inhibitors, where the understanding biology wasn't there. The next evaluation of osimertinib, recently presented just a couple of months ago at ESMO this year, was comparing it to the first generation of EGFR inhibitors in the first-line setting, so in patients who were treatment naive, who had EGFR mutation. And you can see a large study here, the FLORA study, was presented looking at patients with EGFR mutations, stratifying for the type of mutation that they have, randomised 1-to-1 between osimertinib or erlotinib or gefitinib. Here you can see the primary endpoint, which is progression-free survival, and you can see again a dramatic improvement in progression-free survival of 8 months compared to the standard of care new first-generation drugs, and a hazard ratio again of about 0.5. So this clearly demonstrates that the disease is controlled longer with this new class of drug. What's going to be quite interesting is when the overall survival data matures to see how that impacts on overall survival, because of course the sequencing of treatment is not clear just purely from that progression-free survival. However, this data is still very compelling. and has led to the license of the drug. What's also quite interesting is because the osimertinib was developed to be particularly targeting the mutated EGFR and sparing the wild type, therefore it's actually less toxic, or at least has a different toxicity profile to the first generation drugs. And this can be reflected here in the data showing a lower number of grade 3 or 4 toxicities. So now we have a number of EGFR-targeted drugs licensed, gefitinib, erlotinib and afatinib, first and second generation drugs licensed as first-line treatment, and osimertinib licensed now both as a first-line treatment or as a second-line treatment following on from a first-generation drug. So this was really exciting. We had a breakthrough for this subpopulation with the EGFR mutation, but the molecular biologists didn't stop there. The next step was looking at other driving molecular alterations, and the next one found was ALK. And an ALK fusion protein, so this is not a mutation, it's a fusion protein, genetic rearrangement, occurs in 3 or 4% of non-small cell lung cancers. And there's another quite interesting story here in a messages from the drug development of crizotinib. So in this study, the drug was first in human being evaluated in a phase 1 trial. And during the time of the dose escalation, it was recognized in around about 2008, 2009, that there was this subpopulation of ALK-driven lung cancer. And so there was a specific cohort made within this phase 1 study. And the data of this were presented serially at a number of international meetings. And again, here you can see a waterfall plot showing the vast majority of people getting meaningful benefit with a good response and a durable response with progression-free survival of around 10 months, as reflected here in the Kaplan-Meier curve. And this led to the license of the drug in this setting. Similar to the EGFR story, the reverse translation happened where progressing disease, the tumours were biopsied to understand the mechanisms of resistance, and the situation was slightly more complicated here with only about a third of patients having ALK-driven resistance and other mechanisms of resistance being present in other cases. However, there was still the generation of a number of other new drugs, mostly to some degree covering some of those resistance mutations, but also generally being more potent and more selective and specific for ALK. And the drugs do have slightly different characteristics, but here's an example of 3 of the drugs which are most advanced in development. And you can see that in patients who've already responded, been treated to and developed progression on crizotinib, you can see response rates of around 80%, so really active in that setting. And durable responses as well, so ranging from around about 8 or 9 months to over a year. You can see that, again, giving these drugs in sequence, we're getting really good durable response. So this led to data which is presented again just earlier this year, not long ago, of the ALEX study, which is looking at alectinib, which is a new generation of ALK inhibitor, which was developed not only to be more potent against ALK, but also it's a lipophilic molecule which is designed to cross into the blood-brain barrier better to get better penetration into the brain. And this again is a simple randomized 2-arm study comparing alectinib, but rather boldly against crizotinib, which wasn't licensed in this setting at the time, for patients who are naive to all treatment, who have the ALK alteration. Again, with progression-free survival as the primary endpoint. So here we have the data for the progression-free survival by the independent review committee, and you can see that even though the control arm is with crizotinib, which is such a powerful and useful drug in this setting, we see a dramatic improvement with a hazard ratio of 0.5 and a more than doubling of progression-free survival in this patient population. So we're really seeing a massive change in progression-free survival. The data is still very young, and we haven't got overall survival data yet. So we now have a number of other drugs, ALK inhibitors, which are currently licensed and available. Crizotinib, both as pre or post chemotherapy, and then other drugs, alectinib and ceritinib, either post crizotinib or pre-crizotinib. And there's quite a lot of discussion going on about how we should best sequence these agents, but also a number of new drugs coming through. In that first phase 1 study with crizotinib, as well as the ALK translocation being recognised, there was another subpopulation of lung cancer discovered, which were driven by a ROS1 translocation. And again, a subpopulation was generated within this phase 1 trial with about 50 patients. This is a rarer population, about 1% of non-small cell lung cancer. Those patients have a similar characteristic to both EGFR and ALK, being light or never smokers, adenocarcinomas, more common in women. And you can see here again the waterfall plots showing dramatic responses in the vast majority of patients with a number of complete responses. And again, this response is very durable with progression-free survival of more than 18 months. So again, really dramatic effects from this phase 1 study, which has led to the license of this drug in this rare patient population. So BRAF and the MAP kinase signalling pathway is another signalling pathway which is very important in cancer. The inhibition of this has been most established in malignant melanoma. But there's also recognised a smaller population of non-small cell lung cancer also harbour this mutation. And so a study was developed, and the most recent presentation of this again was just a month or two ago, early this year at ESMO, looking at the combination of dabrafenib, which is a BRAF inhibitor, with trametinib, which is a MEK inhibitor signalling the same pathway. And the trial was designed, first of all, with a single drug in the relapse setting, then combinations of drugs in that setting, and then moving forward to first-line treatment in a small cohort of patients, which is what was presented this year. Now, interestingly, the patient population is slightly different here. In this population, patients are more likely to be smokers, or at least they don't seem to be so heavily predispositioned to the never smokers. So there's some differences here in the patient population, but no difference really in the dramatic benefits seen. So again, in this waterfall plot in a very small study in the first-line setting, we can see that almost all patients had some degree of benefit, with a very high response rate and some complete responses. You can see that response is very durable again, very typically around about a year, like we've seen with many of these different targeted therapies within the subpopulations selected. And this has led to the license of this combination of drugs. So you can see here a rather complex slide, which is showing that those 4 examples which have been updated this year with different drugs in different populations, involving reverse translation to try and develop better drugs in that setting, are actually only the ones which we can target at the moment with licensed treatment. But there are trials ongoing looking at multiple other subpopulations, including MET, including RET, including ROS1, we've mentioned already. Of course, KRAS, one of those bigger subpopulations, is a situation where we haven't yet really found the answer to. But this is really exciting, and there's lots of changes going on. One of the similar, better understanding of the underlying biology is seen in squamous cell lung cancer, but the advances in drug treatment to target these drivers has been more of a challenge. In fact, it's quite challenging to do drugs trials in this situation for most patients, because what was started off as a very common cancer, lung cancer, becomes quite rare if you start looking at very small subpopulations. And so there's been a number of strategies which have been developed to try and do this more effectively across different countries, often large national studies with quite complex adaptive designs to try and capture these different changes within a single study. So in summary, we have now a number of licensed treatments linked to particular subpopulations of patients with lung cancer, showing really dramatic improvement in their outcome. And it's really important that we continue to explore further subpopulations and also use that reverse translation to be able to understand better ways of treating these tumours serially as they evolve through time. So this is really important that we do this to try and improve outcomes for our patients.